distinguished speaker, Dr. Jeevan Kumar sir, an accomplished medical genetist with over 18 years of expertise in clinical cytogenetics, molecular genetics, next generation sequencing for rare disease research. Sir currently is serving as an assistant professor and assistant director of academic affairs at the Apollo University, Chittoor, where he plays a pivotal role in shaping the biomedical education and advancing research in human genetics. His academic journey includes a PhD in biomedical genetics from VIT University, and his career reflects a perfect blend of teaching, research leadership, and clinical application. He has been the principal investigator for multiple funded projects, authored numerous high-impact publications in journals like Aging Cell, which has an impact factor around 8, and European Journal of Human Genetics with a good impact factor of 4 to 5. He has also delivered, he has been invited as a guest speaker and has given a talk at various prestigious forums such as ATAL, ATAL, FDP, and PIGS. Sorry. He's been recognized with the Best Researcher Award at SRMST. Dr. Jeevan Kumar's contribution extends beyond research to mentoring future scientists, promoting genetic literacy. Today, he will share his insight on pedigree analysis of rare diseases, a subject where he is an expert and passionate about. Please join in giving a warm welcome to Dr. Jeevan Kumar. Thank you.
Very good afternoon to all of you. Thank you, the organizers, Shana, Jeevan. Thank you for the elaborated introduction. So it is indeed a pleasure to deliver this talk as part of the workshop titled Pedigree Analysis of Rare Diseases. Before we start, as part of the workshop, I was asked to give a pre-test. So I request all the participants to take a scan of this QR code. There will be 10 questions, multiple choice, simple disease to complex disease. Okay? So that's the main aim of this workshop. As Madam briefed, this is my overall journey from 2006 from Children's Hospital. By March 2026, I'll be completing 20 years in this area of medical genetics, where I handle diagnostics, research, and teaching, which is my passion. Across this institution, these are not a separate institutions. I have my own dots. I started as a cytogeneticist, then entered into an area which I don't know, and then before joining Apollo, I joined before that I was working as a postdoctoral fellow in rare disease genomics. Before joining postdoc, I have no idea about genomics. I learned a little bit after joining KMC for 3 years at a DBT project. Okay, so with this background, I thought I want to share some of these pedigree analysis. It's not just overnight slides. I have got these slides over 15 years of period. I just want to highlight these points as part of this workshop. I will give a brief on pedigree. Pedigree symbol, patterns of inheritance. We have two patterns of inheritance. One is Mendelian inheritance. The other one is non-Mendelian inheritance. We call it as a traditional, non-traditional inheritance. Then I want to highlight what are the challenges that we face when we see a pedigree interpretation, when we try to interpret a pedigree. And last, I want to give a case study where I want to emphasize the importance of doing pedigree analysis for one of those rare cases which we have seen in the clinic. So, two things I want to highlight. One is the pedigree, the other one is the rare disease. As you know, rare disease has an incidence of 1 in 2,000 individual, where the incidence is for a disease, what we call it as a rare disease. And over till date, we have about 10,000 rare diseases in human, which is being discovered, and the genetic basis is already known. And over 70 to 80% of these rare diseases have underlying single gene issues, single gene mutations that is contributing to the disease. And as we know, many of these genetic diseases has no cure. This increased risk for morbidity and mortality. And over 90% of these diseases has no approved drug. So we are working with a condition predominantly focused for prevention and screening. We have little help from the National Policy for Rare Disease 2021 from Government of India. We have about 10 crore people suffering from this rare disease in India. And on average, the time taken for diagnosing this complex disease is minimum 4.8 years, maximum 20 years of time to figure out the underlying basis for rare disease. And 40% of these diseases, when they go for the first consultation, they give a wrong diagnosis. That's the experience with rare disease. Okay, so coming to the second part of my talk is pedigree analysis. So pedigree, which is a Latin word, originated from Latin. The word which means pes refers to foot, the gross refers to crane. So if you see the horizontal lines, vertical lines in the family tree, they look like a crane foot, hence the name pedigree. So when I posted this pedigree workshop, one of the faculty asked me, "We're talking about the dog's food, pedigree." So they know this. So then I said it's about family trees. And pedigree analysis is nothing but a shorthand representation of the medical history of a family. So, anybody know him? He's the first person who gave the square and circle for male and female in the pedigree. He used the first symbols for male and female representation of the pedigree. So as we know, the open circle is male, it's for basics. Open circle, square is male, circle is female. And before we learn pedigree, it's always better to know what and how pedigree can help someone who is practicing genetics. So first of all, pedigree helps in making a diagnosis. It also decides on the type of test that you order, whether it is a chromosomal test, single gene, or complex a genome test. It also helps decide who to test, identify patterns of inheritance. It also helps you to calculate the risk of recurrence of the disease among family members. It also helps you to make a repertory option for the family who is suffering from genetic diseases, distinguish genetic from non-genetic causes of the condition. It also helps make a decision on management and surveillance, like the cancer, where we do a surveillance for a long period of time. So develop a rapport between the patient and the doctor. It also helps to educate patients and families about the rare diseases. So these are very basic patterns or symbols that we use to denote and use to construct. The reason for using the standard nomenclature is universally we follow the same language. So for example, these abbreviations, for instance, I will tell you an instance. where there is a termination of pregnancy, for example. So we use a shorthand form as TOP. Some will write MTP, medically terminated pregnancy. Some people will write TOP, termination of pregnancy. So across we use different ways of representing these things. But there is a standard nomenclature where universally we follow the same language to construct pregnancy. So to start with, we have the open square, open circle. We know already they are denoted for male and female. When there is gender is not known. So for example, if there is an ambiguity in the gender, then we use a diamond-shaped symbol to represent pedigree. Then if you shade them, it is affected. The individuals are affected. If you give a multiple shades, that means that individual is having more than one disease. We usually give a multi-shades, that means more than one disease. This individual has more than one disease. Sometimes what happens is the pedigree, when you construct, it will be full, page will be full. So for you to have a shorthand representations, we use either number of names. For example, here in this instance, it is written an open square with 5. That means there are 5 male individuals. So we can accommodate the pedigree within a sheet. So there is always a rule also to follow when we are writing, when we are constructing pedigree. We should always use white paper. Number 2, we should use black pen. And we should always start in the middle of the page, so that we can extend on either side, male and the female side. Always, male, the father should be on the right, mother will be on the left. This is a regular pattern followed across institutions. So here we can represent 5. That means there are 5 male individuals. This 5 female individuals, and this is 5 individuals. Sometimes what happens is the family member says that there are 10 people. I don't know how many are male, how many are female. So that case, you can just say the gender is unknown, but the number of individuals are 5. If they still don't know the overall number, they don't have the exact number, then we can write n, small n. Number is unknown. And across the square, circle or diamond, that means the individual is died or deceased. And when you put an arrow to an open circle or square, that means the individual is a consultant who comes seek advice from a consultant. We call them as consultant. So the T is the difference. And this symbol, I think everybody knows, this is a program. We have to denote by capital P. That means the individual is affected and through whom the family came and seek advice. So this is an affected individual. And to denote stillbirth, we have to write SB and a slash. We have to write the number of weeks of gestation that denotes stillbirth. This holds good for all the male, female, as well as the gender unknown. And pregnancy is represented as capital letter P inside the square or circle or diamond shape. Usually we don't reveal the gender. So what happens is we use a diamond shape and then write a P. That means a mother is pregnant. And we need to always write the number of gestation weeks to denote the pregnancy, ongoing pregnancy. And then spontaneous abortion is denoted as triangle. If you shade, that means the individual is spontaneous abortion with a defect. If you just keep it open, that means no defect. This is spontaneous miscarriage or abortion. Termination of pregnancy, we need to slash and then write the cause. If you know the cause, then we have to shade and write it. Here in this case, it is 47XY plus 18. It's a chromosomal defect. What's the name of the condition? Everybody? Trisomy 18. Edwards syndrome. Okay. So Edwards syndrome. We need to write the diagnosis, and if it is affected individual, we need to shade. Ectopic pregnancy is denoted by ECT. abbreviation underneath the termination of pregnancy, because many times pregnancy will not go as a complete pregnancy, right? So ECT represents ectopic pregnancy. If it is terminated, we have to put a slash.
What is the square in shape, sir?
Then,
Ninth one.
but there is a karyotype abnormality 47XY plus 21. So the fetus is found to have trisomy 21.
It's a male fetus.
It's a male fetus. Male fetus.
Only your fetus
Yes.
has this.
With a diagnosis. And in constructing pedigree, we use horizontal lines, vertical lines. So each line represents a unique meaning. If you join the male and female with the horizontal line, this line is called relationship line, or they are in relationship, or they are married. If you draw a vertical line from the relationship line, it's called line of descendant. And to the line of descent, if you write one horizontal line, then it's a ship line. And from the ship line, we have these individual lines. So each line has a unique definition. And then, if you cross the relationship line with a double line, that means they are separated. They are divorced or separated. And if you put double line between male and female, then the individual, the marriage between them is a blood related marriage. Called as a consanguineous marriage. So over 50% of marriages in South India, especially Andhra, Karnataka, Tamil Nadu, where the marriages, first cousin marriages are very common. The percentage now is going down, like now it is around 40%, but still first cousin marriages are very common. And if you, this is a relationship line and the individual line here, biological, which denotes the biological parents. If you join the individual line here, that means the individuals are monozygotic. If you don't join them, that means the individual is dizygotic. If zygosity is not known, then we put a question mark. That means the twin zygosity is unknown. We are in the era of assisted reproductive techniques, so triplets. Pregnancies are very common, so to denote that, we use this symbol, trizygotic pregnancies. And then, if the family history is not available, if the individual is not able to disclose or not able to know the family history, we just put question mark. And to represent no child by choice, we put one line. For infertility, we put double line. So there's always a criteria for infertility. More than 1 year of marriage life, if they don't have kids, usually they come under a group called infertility. So that you can denote by double line. If there is an underlying reason, then we have to write, like angiospermia here in this case, endometriosis. So if the condition is known, we have to write the cause for the infertility. And for adoption, you can see this line. What is this line for? Relationship line, line of descent, ship line, individual. And here you can see dotted lines. That means these individuals, this individual, the parents of this individual, they are not biological parents of him. So there is a dotted line. If you draw a solid line, that means they are biological parents. So here we also represent with the big brackets, which means they have adopted this boy into their family. And they are not the biological parents, and this boy is adopted into the family. So somebody gave the boy out, adopting out, then we can give a solid line and we can represent with the brackets. Sometimes the relative themselves will adopt in and they adopt out, so that we can represent here. So here what happens is the mother adopted son from her own sister. Okay, so she is adopting in and she is adopting out. she's adopting out, she's adopting in. So she is adopting out, she is adopting in. And this is for symbols represented for assisted reproductive techniques, ART. So the first top one is the sperm donor, gamete donors. So when you put a square in capital D, and this conception is through a donor sperm. If you put double line, that means the couple is having underlying infertility, and they went for donor sperm for the pregnancy. And this is for home donor. So sperm cell from the father, the egg from the donor. So we have a pregnancy here. And then this symbol represents the surrogate mother. We have the cells from the parents, and we have taken this division for surrogacy. And then this is for surrogate home donor, where this individual donated egg cell, and then also as an individual for surrogacy. And the last symbol that denotes this couple went for an adoption using donor parents. And this is a last slide that denotes these symbols. If you put an asterisk marks, that means the individual is evaluated by you as a clinician. If you put a dot, I think many of you know this. This to represent the carrier status of the individual. If you put a thick line in the center of the individual, that means the individual has a variant, has a mutation or underlying genetic basis. At that time, is not having any features. So we call them as asymptomatic mutations. The best example is if you identify a BRCA mutation in the individual, for her the risk of having BRCA mutations with the ovarian or breast ovarian cancer is quite common. Or maybe the instance is like over 80% of them have a risk of having breast ovarian cancers. So that time when you evaluate, she may not have any features of the clinical signs. We call them as asymptomatic mutations. So to represent that, we have to use this solid line in the center, which means asymptomatic individual. And when you have a diagnosis, we have a test done, but still we cannot able to associate the findings. Best example is the Huntington disease. So Huntington disease comes under a group of disorder called triplet repeat expansions. So there's a particular type of triplet repeat which code for a specific amino acid called phenylalanine glutamic acid. So what happens is we all have the repeats for about 36 or 35 repeats, minimum 24, maximum 35 repeats. Someone they have a 36th repeat, they either have a risk for having Huntington disease, or they may not prone for Huntington disease. So it's a kind of a gray area. We don't know whether they will have the disease or they may not have the disease. So to represent that, we use this symbol, uninformatic study. Study we have a finding, but we cannot able to conclude using the findings. The last one is the E plus, which represents, it is evaluated by you, and plus represents a positive diagnosis. So here in this case, you can see this carrier parents. So you have a dot here, that means a father is carrier, mother is carrier. Child is a homozygote recessive condition here. And you can see delta F minority mutation. So this is one of the common mutations that you see in cystic fibrosis, where they have, this is one of those recessive conditions, autosomal recessive disease. So to represent that, if there is a positive test report, we represent that by plus, and E represent the evaluation done by them. So here in this case, we have a fetus with the P stand for pregnancy. The mother is pregnant with a male child with 10 weeks of gestation, and E plus represent the evaluation is done. Test is done using the CDS sample, sample, and they figured out that the sample has 47XY plus 80, which represents an Edwards syndrome. So when we have a positive diagnosis, we have to represent that using this symbol called E plus.
So the arrow mark that you showed in the previous one, that will be given for the mother in that case. If the P that represents the baby,
yeah,
that means the arrow mark that you said before.
yeah.
No, before. The last. That arrow mark will be for the mother.
This is for the individual.
No, just the arrow mark. The arrow mark is the consultant, right?
Yeah, arrow mark is a consultant. We also use arrow mark to denote program. When you write P, that means program.
Yes.
When you don't write P, it is consultant.
So when you are putting that one where the square with the P, that's the baby, that's a fetus. So the consultant will be the mother.
Consultant is the mother, of course. Mother.
So the arrow mark goes on the mother.
Yes. So this is basically to denote the underlying problem of the fetus. Fetus is affected with the chromosomal disease. So this is a typical sample pedigree. So quickly we can come compose this basic pedigree symbols. So can anybody quickly tell me what is this symbol stand for?
That's it.
This one? Deceased. This?
Affected and consultant. Proband.
Proband. I didn't write the P. This one? Mother is pregnant.
Pregnant, gender unknown.
Gender unknown. 24 weeks. This one?
That is number 3, right, sir?
Listen, I'll tell you.
No, sir. When you say P, that is P, that means that number 3, the mother, that person is pregnant.
No, this.
That person is pregnant.
Yeah.
Okay.
This is the line of descent, kinship line, individual line. So this couple already had one boy who's a proband, one normal child, one termination of pregnancy, cut ongoing pregnancy.
What is proband, sir?
Proband is an individual who's having a condition.
Who is affected?
Affected. Generally they are affected.
Affected individual is called proband.
Sir, affected individual who is the consultant is the proband
To whom the entire family come and seek advice.
Consultant who is a positive.
The consultant need not be always affected.
No, sir. The proband means a person who consulted and also positive.
Positive, having the condition. Having the condition.
No, it depends on if it is colored, then it is diseased.
Affected.
Affected.
So if
Because
it is
just the arrow refers to consultant.
yes, sir. If it's a square with full shade, sir, that
Yeah.
means he is affected. But if the full shade square with the arrow means he is the person who consulted and also affected means he is a proband.
Yeah, we can say that. We can say that. Yes.
Correct, sir. I mean, I am also getting it. Yes. Yes.
So coming to the consanguinity, as we go very close to the relationship, usually the amount of genes that we segregate is quite high. So for example, the percentage of genes that we, say for example myself, the percentage of genes I get from my father and mother is 50% from my father, 50% from my mother. If I take 3-4 generations from after me, then the same percentage of genes that I inherit to the 4th degree or 4th generation, 5th generation, the percentage is less. So I can give this example with a very simple analogy. If I give you a coin, 2 coins, if I give, the percentage of getting you head or tail is 50. 1 coin, 50%. 2 coins, 25%. If I give 10 coins,
5%.
the percentage will go down. Maybe high or it may go down. So the probability of inheriting the defective genes down generations will go down. So if you are so close to the relationship, so for example, the top one will never exist in you. Father marrying son or daughter, father marrying daughter will never have and siblings marrying themselves will never happen. Only happens in animals only. But so what happens is the percentage of genes that they share for their kids will be high. If you go down to these away from the generations, for example, this is the first cousin marriages. It's the first cousin. You can go to the first cousin once removed, second cousin. You are far away from the relationship. So your percentage of sharing the defective gene will go down. Percentage-wise, will go down. So always after fifth degree of consanguineous marriages, it's considered to be a normal population risk, general population risk. So this is called consanguineous unions and the risk of transmitting the disease genes. So you can see here for a first degree relationship, the proportion of gene inherited is 1/2. For the second degree relationship, 1/4. Third degree relationship, 1/8. Fourth degree, 1/16. Fifth degree, 1/32. So as you go away from the relationship, the percentage of you getting those defective genes is less. And by the way, in fact, irrespective of the gender, we all carry 3 recessive mutations. So by nature, we are all mutants. Even though you say that we are normal, but we all carry 3 recessive mutations. That means on every replication, we have about 3 billion nucleotides to get replicated for every cell division. So per replication, we harbor 3 mutations. So that is the efficiency of our replication. So these mutations are generally considered to be recessive ones. And to give a summary, can you be a little louder?
The last ones,
Uh-huh.
we take those vertical lines as individual lines or the descent lines?
We get from the descendant. These are descendant. These are not individual lines. It's a line of descent. All lines are descendants. But this first one is a line of individual line. From then the offspring. For them this is generation 1, generation 2, generation 3, generation 4. So in summary, these are the abbreviations we should commonly follow for pedigree construction, and these are all the definitions for each and every abbreviation that we use in pedigree. Whenever we evaluate pedigree, these are the points that we should always consider. Number 1, how many number of times a particular condition is occurring in the family, which generation is commonly affected, whether disorder is maternal side or it is coming from paternal side, age of onset of the disease, age of death, gender of individual affected, is there one type of disorder running in the family, any clustering of diseases, the nature of those clusters we have to figure out whenever we see any pedigree chart. And these are the questions we should usually ask while constructing the pedigree. This is called the red flags. We have to ask, is there any chromosomal disorder, intellectual disability, speech delay, deafness, vision problem, and then they have attention deficit hyperactivity. So all those common conditions running in the family we should inquire. So first we will start with the regular pattern of inheritance. Many of you are aware about these pedigree charts. I just want to show you. So these pedigree patterns, there is a clue. Next time if you see pedigree pattern, the way you look at these pedigrees will be different. So there are clues. For example, I am calling this as autosomal dominant inheritance. They have a characteristic patterns. So you see, there are how many generations of pedigree is this? 1, 2, 3. So always we should have 3 generation pedigree. Positive criteria. Number 2, who are all affected here? Both male and female. Who is transmitting the disease? Female to male, male to female. And you can see there is a vertical transmission of the condition. So whenever you have both gender affected, all individuals are affected, I mean both male and female are affected, there's a male to female, female to male transmission. Close your eyes and say autosomal dominant pattern. So this is a classic pattern of autosomal dominant. And you see, if there is a affected male, for him there is a 50% risk that either his boy or a girl will have the same condition. So overall percentage of having the condition is 50%. So consider this is father. Father has 2 copies of genes. For instance, he has one defective gene. I put capital letter D as a defective, small letter as a norm. Likewise for mother, say you consider mother is normal. Small letter. Okay. So what type of gametes you can expect from this father? There are 2 types of gametes. One is capital letter D, the other one is small letter D. Mother excels normal gametes. So this is sperm cell, this is an egg cell. So when you have these gametes, the combinations will give like this. So overall, what's the proportion? We have 50%. Father is also affected with a defective gene. So 50% of his offspring will be affected. 50% will be. So it's a classic autosomal dominant pattern. Why it is called autosomal dominant? Why it is autosomal dominant? Because these genes are located in the autosomes. 22 sets of chromosomes. If they are the sex chromosome, we call it as sex-linked inheritance. Okay, so this is a typical way we can either write like this segregation or we can put Punnett square. I think Punnett square many of you are aware. Just for the beginning, I just want to show. So for Punnett square, so this side for male and this is a female. You have a capital letter D from the father who is having a disease. Small letter D has a gamete, and mother with the small letter D, small letter D. And this combination will give and you have. So either you can represent as segregation. For first instance, I want to show this. Later, I will want to repeat this. So 50% of this offsprings from this father will have the same disease. Okay? So this is a typical representation of autosomal dominant picture. This is segregation. So you can see father with the capital D, small d, mother normal. So you have a 50% overall. Wherever there is a capital D, they are affected. Small d, they are. So 50%. So quickly, these are some of those cases which we have evaluated in the children's time. Clinicians are there. Anybody with the diagnosis? Syntactically, radiostenosis is a condition called Apert syndrome. Apert syndrome. First, this is a child with achondroplasia. This is a child with Treacher-Collins syndrome. This is a child with neurofibromatosis. There are variable features in neurofibromatosis. And see, this is a condition called dentinogenesis imperfecta, which is part of the OI, osteogenesis imperfecta. Same family member, they have variable features. Mother will have just a teeth anomaly, child will have fractures. So it's one of those severe brittle bone disease that you see called osteogenesis imperfecta, and this is cranio-cranial dysautosis. Usually they don't have this collarbone. It's one of those features of cranio-cranial dysautosis. Coming to the recessive pedigree, I know many of you know this picture. Yes?
Egypt.
Egyptian Cleopatra. This is a Cleopatra's family tree. So Cleopatra married her brother. This is some of those unusual pedigrees that we have in history. Any guess? This is a French artist, painter. They have a tradition of the family fond of painting. They're also fond of drinking alcohol, so that they kind of live those French lives. French artists, they have a strong tradition of artistic and fondness for alcohol. The greatest artist, his name is Henry, and the parents of them, their first cousins. And then he got a very rare disease called pycnodysaustosis. It's also a type of bone disorder. They have short stature and fragile bone. It's Henry, and he got a rare disease called Autosomal Recessive pycnodysaustosis. And whenever you see now recessive pedigree, parents usually are carriers, and you can see here which generation of individuals are affected. Third generation. So whenever you see a recessive pedigree, individuals are in one generation. Affected individuals are in one generation. Whereas in dominant pedigree, the inheritance is vertical transmission. Recessive pedigree, horizontal. The individuals, affected individuals are in horizontal line. So there is one group for recessive pedigree. And here, so if you see the inheritance, if father and mother is a carrier for a genetic disease, for this instance, we have a chromosome pair. One of the allele is having mutation, the other is normal. Likewise, mother is also a carrier for one mutation, the other allele is normal. When they give rise to the gamete, if the mutant forms come, the individual is affected. If they get one defective gene, one normal gene, the individual will become carrier. And there is also a possibility for both the allele is normal. So overall, what is the percentage? If you have a carrier pattern, 25% is defective, 15% is carrier, 25% is normal or unaffected. So this is a typical pedigree. I just want to show if both parents are carriers, this is the risk: 25%, 50% carrier, 25% affected.
Sir, go back to the previous.
Yeah. This one?
No, previous.
Yes.
No, previous.
Previous.
You told that this is autosomal recessive.
Recessive.
You told that parents are recessive carriers,
Yes.
and third generation will be affected.
If the parents, both the parents are carriers, the risk for having a baby with the defect is 25%. We can draw the same things.
Can we visualize one more
We can do that. Half, I gave half gene is normal, half gene is defective.
One
dot
One
also
dot
is
is
carrier
also
also.
That dot we use for X-linked. I'll show you.
What?
X-linked condition.
X-linked condition.
Carriers.
Carriers are parents, the next generation will be
Effective.
immediate.
Yes.
The next thing what you showed is in third generation. Next slide. That is third generation.
So I'm giving you an instance where parents are carriers. What is their risk of having baby with a defect?
So with reference to that one, this is the third generation.
Yes, yes, yes. Sir, still.
Yes, still
Okay. Sir, we will take like this, sir. We all have pairs of genes. We get one set from the father, one set from the mother. Many times what happens is, we require both the copies to express. We need both the copies should be functional. Sometimes what happens is, half gene is enough for us to have a normal. There are genes, we call them as a recessive genes, where half normal, half defective, we call ourselves as carriers, but we have a risk of getting a baby with a defect. When it becomes homozygous, when both genes are defective. The third category of genes are there, where we have both the copies, but based on the origin of the parent. So for example, you get very strongly one gene from the father, one gene from the mother. Sometimes what happens is gene express when it is present on the father's chromosome. The same gene on the mother is silent. Vice versa. Sometimes the genes on the mother express, the same gene on the father's chromosome is silent. We call this as an imprinting defects, imprinting mechanism, or we call it as epigenetic mechanisms. So this is how our genes expresses. Predominantly we require more genes. Sometimes half gene is enough. Sometimes parent of origin of the genes will have a normal life. Will lead a normal life. So this is how our genes work. So the first category, what I said is, we have two copies, one gene is defective, that means we are manifesting the disease. The second scenario, recessive mutations, half gene is enough, so we can have a normal life. Half gene is defective, still you don't have any problem, but when you give rise to babies, your babies will have a risk of having a homozygous recessive. So here, in this example, what I am trying to explain is, father has one normal gene, one defective gene. Mother has one normal gene, one defective gene. Based on the combination of the genes, because one has to come from the father, one has to come from the mother. If you get the defective gene from the father, defective gene from the mother, it's like a probability. So every time you can get both times heads, sometimes you will get both tails, like a tossing a coin every time. So every pregnancy, this parents will have 25% risk of having defective baby, 50% chance of being carriers, 25% chance of having unaffected conditions. So many times when we counsel families, they will say, okay, first time I got a defective child, second time you are saying carrier, so can I try one more baby? They didn't understand the probability. So every time the risk is 25%. 25% risk is a huge risk. So we always draw them and then explain them. So I will give you one small work. If one parent is carrier, what is their risk of having a baby with a defect? None of them will have. So there is a 50% of the babies will be carrier, 50% defect. One more scenario. One parent is affected.
All are carriers.
100% they become carriers. Okay. So these are some of the examples I thought I want to give so that you will register them. The first case is Ellis Vancouver. It's a condition. They have polydactyly, both feet and hands. This is a condition, polydactyly obesity. It's a condition called Bardet-Biedl syndrome. This is a photosensitive skin. It's a condition called xeroderma pigmentosa. It's a DNA repair. It's a defect. You see here. This is a child with any guess? Name of the disease? One of those storage disorder, lysosomal storage disorder, mucopolysaccharidosis. So these are all come under autosomal recessive pattern of inheritance. And we know the other common condition under recessive disease is cystic fibrosis condition. Coming to the extinct inheritance pattern. So when you say extinct pedigree, usually what happens is females or carriers, males are having the disease. Okay? And in dominant pedigree, you see vertical transmission of the disease, recessive, autosomal recessive, horizontal. In extinct recessive, you see oblique pedigree. Affected individuals are an oblique one. You may see only two males here. When you draw extended pedigree, you will understand better. So you have a father, mother. Say for example, if this mother is a carrier, extinct. So extinct means the mother will have two X chromosome, father will have one X and Y chromosome. So defect in this single X chromosome, father will be affected. So here in this case, I am giving it as a normal X for the father. Mother is having a defective X. One X is defective, the other X is normal for her. So I am talking about recessive disease. As soon as I said recessive disease, one copy is sufficient. The other copy is having a mutation. So she will become carrier. Whenever this defective X goes to the child for a son, he will have the condition. Always father give the Y chromosome to the son, the child or mother will give the X chromosome. And if this female child gets a defective X and one X always coming from the father. So what is the condition of this child?
Carrier.
Carrier. This is 50% risk. 50% risk. 50% of males will be affected. 50% of male has a chance of getting normal X and one Y chromosome from the father. So what is the condition of this child? Normal. So 50% of the males will have the disease. 50% of the females will have become carriers. So in this we can see an oblique pedigree because the individuals, affected individuals are males, and we can draw one diagonal line. So if you just extend this pedigree, father with the brother and her, mothers, brothers. So when you see this pedigree, the affected individuals are already oblique line. So these are another characteristic feature of one of those interpretations for extinct recessive. So in Victoria's family, we know there is a strong history of breeding disorder or hemophilia. You can see who are all affected in this pedigree. Males. So because it's a typical extinct recessive pedigree, the defective X chromosome in the male usually manifests. So why females are not expressing? They have one X normal, the other X is defective. Since they have one X chromosome normal, they can become a normal individuals. Or they become a carriers. If they have defect, they will become a carrier. So this is the same segregation which I have explained here. So you can see the father is normal here, mother is having the mutation, small letter R, the highlighted with the red color. So wherever there is a red color, with the Y chromosome, X and Y, the individual is affected. The XX with the defective X, then the female will become carrier. So overall, 50% of females will be carriers, 50% of males are. So we can try that in the honors.
Can we know which order? In the first child will be affected or second child will be
That's what I'm saying. On every generation, you have risk is either male or female. But overall, in the pregnancy, the risk of having a boy or a girl is 50%. So for a boy, if it is a boy, 50% of the boy will have recessive. If it is a girl, 50% of the girl will have become a carrier.
Yes, yes.
So overall, it is.
Sure.
Okay, so this is a recessive pedigree, characteristic features I have given. Sometimes females will also get recessive condition. This is very rare. We can consider when we have a female affected with the recessive disease, where the father will be affected, mother will be a carrier. So this is one of those rare possibilities.
Sir.
I'll go step by step. We have father, mother, and what I said is father is affected. That means he has one X chromosome, one Y chromosome. X chromosome is having the mutation. So what's the disease state? Affected. Mother is a carrier. One X is carrying the mutation, the other X is not. When you have parents like this, mother is a carrier. So what type of gametes we can expect from the father? Father, X and Y. X is having the mutation. Mother, one X is defective, the other X is normal. So when you have a gametes like this, this is father, this mother. So what combinations you get? X, X, both are defective. This X and this X. This X and this Y. Y and X. So what is the status of this individual? Female, affected female. Carrier female. Affected male. Normal condition. So, is there a possibility of female getting extinct recessive disease? Yes. When the father is affected, mother is a carrier. Very rare possibility. And we have individuals where who have Turner syndrome, 45X. They have only one X chromosome. If that is defective, they manifest the disease. So I have seen two individuals with extinct condition called DMD, Duchenne muscular dystrophy in a female. So female with DMD is very, very rare. We had one case with the sex reversal with a female, but she got 46XY character. There's a condition called Swyer syndrome. I think Rajesh is here as well. So there's a condition called Swyer syndrome, and one condition we saw is the X autosomal translocation, very rare possibility. The female had DMD, and they confirmed that the DMD gene also got deleted in one of the X chromosomes. So this is the possibility when you see when a gender doesn't unusually has a rare extinct recessive pattern, we have to consider one of these possibilities. These are some of the examples. We know already DMD, BMD, G6PD, hemophilia, Menkes, lesion in hand, Fabry, red green color blindness, or some of these conditions. And I have given the names of these genes and the location on the X chromosome. Okay, so coming to the X dominant type, you already will answer. So what is this symbol stand for?
Still good.
Spontaneous miscarriage.
Still okay.
I have not shared it with you. So it's a spontaneous miscarriage. What is this symbol?
Affected female.
Affected
Female.
female individuals. So From this pedigree, what is your observation?
It's Y-linked.
Females
Sorry.
predominantly are affected. In males, the condition is lethal. Even formation of male individual is not happening with the disease. So whenever you see a pedigree where there is a male lethality, when the survival will never happen with the disease, we call it as a lethality. Okay, that's a lethality. So whenever you see multiple male spontaneous miscarriages, affected females in the family, possibility of X-linked dominant disorder is likely. Okay. So, similar way we can structure the Punnett square. If you have a mother who is, usually what happens is this kind of conditions where one defective gene is enough to manifest the disease. Hence the name dominant. We said dominant disease. So the risk of having a baby, in this case it's a female who's with a defective gene, so she will manifest the disease. So here it is 50% of females will have the disease, 50% of males, usually the condition is lethal or severe in the males. So these are some of these conditions where we see X-linked dominant pattern. This is a condition where ectodermal dysplasia, rare form of X-linked dominant. Sorry, this is a recessive. So this is a recessive condition. So I just want to give the beginning. So I just want to show you. This is a boy with a DNB, X-linked recessive condition, where you can see hypertrophic heart cough muscles. These are some of the examples I thought want to give for X-linked recessive. For X-linked dominant, I want to give these conditions: Fragile X, ornithine transcannabinoid deficiency, vitamin D resistant rickets, and lethal conditions in male, orofacial digital syndrome, incontinentia pigmenta, and Rett syndrome. So usually Rett syndrome will have a behavioral issue. They will have a hand waving movement. Usually you will see in females, no male with Rett syndrome, because in males it's lethal. Formation of baby with the Rett syndrome will never happen. So coming to the other pattern, Y-linked inheritance. Y chromosome is one of the smallest chromosomes.
So for X-linked recessive gene, there is a carrier and affected, but for an X-linked dominant, there is only affected and dead.
Yes, lethal.
There is no carrier in that.
No carrier. Because as to they have the defective gene, they will manifest. Dominant is like that. Whenever there is a defect in the gene, they will manifest.
So dominant X-linked means there is always an affected female and terminated or lethal male. Males are always
severe
dead.
in males or lethal in males.
Okay.
So coming to the next pattern, Y-linked inheritance pattern. Y chromosome is one of the smallest chromosomes that we have. Got already 49 genes. The dominant role of Y chromosome is decide the gender of the individual, male gender identity. That's the only function. Yeah. So, and they are also involved in the spermatogenesis. Usually, Y-linked inheritance we see from the father to the son to their son. So you can see that there's a vertical transmission, and it's always inherited to the father. As father will give the Y chromosome to his son, right? So we have a 4th-generation pedigree. You can see a dominant kind of a vertical transmission. But you can see affected male or affected father, all his sons are affected because Y chromosome anyway has to go to the male. So conditions commonly we see under Y-linked condition is SRY gene deletion. It's one of the critical genes for the sex determination in male. And YQ deletions usually associated with the agispermia. We have Y microdeletions. Syndrome is one of the common features that we see in Y-linked conditions. So far we have covered autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked. So any questions if you have in the traditional method of inheritance?
So seniority depends whether it is homozygous or heterozygous case. Rest of all is
Recessive means heterozygous carriers. Dominant means the carrier There won't be a carrier. It will manifest. One single gene defect will manifest. So, just a recap: autosomal dominant, vertical transmission; autosomal recessive, horizontal, X-linked, oblique pedigree, X-linked dominant, lethal in males, severe affected females. You can see in the pedigree. Coming to the non-Mendelian inheritance pattern, we start with the multifactorial disorder, where both genes and neurological factors play a role in the cause of disease. This is one of the common diseases that we see in human. As a genetic disease, this group is very common. 30% of 25 to 30% of genetic diseases will come under multifactorial disorder. So if you see multifactorial disorder, usually we can see the distant relative is affected. With either the same disease or different form of a disease. So for example, you take an example of spina bifida or the open neurotic defect. We will have a different pattern in the previous generation. They may have anencephaly. This program may have a spina bifida. So it's like a variable presentation you can see, and we don't see any vertical transmission, horizontal transmission, or oblique pedigrees. So both genders are affected. Recurrence risk is very low. Prediction is usually based on the empirical risk data. I'll tell you what empirical risk data is. So these are some of the examples I thought I want to give. So spina bifida, anencephaly, cleft lip, and cleft palate. So they all come under a group of conditions called multifactorial. So whenever you see a multifactorial disorder, severe disease in the index case that you can see. Index case is the first person. You don't see any other individual in the family will have the disease. Only one person will come with the disease. So we call them as an index case. Index case being sex not usually affected. That means if you see that only male is affected, it doesn't mean that always males will have it. Even females will also have it. As you become very close to the relationship, more risk of having the disease. Consanguinity plays a role in bringing the incidence of disease is high. There's a more chance for heritability of these conditions as well. So empiric risk data is usually what happens is they adopt data done other ethnic groups, and they apply for the similar kind of conditions in other groups. So to give a brief example, so here there's an empiric risk data given for cleft lip condition. If the individuals are identical twins, that means they share similar genes, so the risk of having disease is very high. So if one twin is having cleft lip palate, the other twin has 40% risk of having the same. If one sibling is having it, the risk is 4.1% for the next sibling. One child is having, second child, there's 3.5%. So niece and nephew, for the next person in the individual, that means you or your offspring, the risk is 0.8%. First cousins, 3%. General population, 1%. So as you are away from the relationship, the risk of having multifactorial disease is less, because we don't share the closest infecting genes. So coming to the other non-traditional method for pattern inheritance is mitochondrial condition. As we know, mitochondria has a circular DNA, is defective in these genes. Also, DNA will also manifest a type of genetic disease called mitochondrial disorder. So, basic feature of mitochondria is that mitochondrial DNA doesn't undergo crossing over, no DNA repair, always maternal inheritance. So they inherit only from, all of us have mitochondria that is exclusively we got from the mother. Father's sperm cell, the mitochondria being away from the head region in the neck as a repulsion mitochondria that just helps in the movement of the egg. So majority of the mitochondria that we make is from the mother, cytoplasm. And many copies per mitochondria per cell, high exposure to oxygen free radicals inside the mitochondria, no histones, no introns. This is a general feature of mitochondrial DNA. So there are conditions that usually found associated with the mitochondrial inheritance is liver heredity of very few. Now people are now started associating with anywhere from migraine fever, migraine headache to diabetes also. Now they are saying that mitochondrial DNA is critical for diabetes. We don't know still. So cancer, chronic progressive external ophthalmologia, mitochondrial encephalopathy, rapid fiber, neurogenic weakness, nerve, brain. These are all part of mitochondrial DNA damage. And if you see mitochondrial inheritance pattern, can quickly tell you who are all affected in this family. Both gender is affected. So there's one small clue here that differentiates. It also shows a vertical transmission, but there's one difference. Can you figure out what it is? Anybody? You have seen autosomal dominant pattern. There you see vertical transmission. Both male and female transmit the disease. But there's a small difference.
Males are not transmitting. Males are not transmitting the disease.
Exactly. Because you get mitochondria only from the mother, not from the father. So when male is affected, male will never transmit the disease to the generation. So this is one difference that we can see whether it is a dominant, autosomal dominant or mitochondrial disease. So this is a classic pattern that we see here. So always this condition is a matrilineal. There's mother alone transmit the disease. Male do not transmit the condition. Both sexes are affected. Risk estimation is usually a difficult scenario because mitochondrial number varies according to the functionality of the cell. So brain cells will have more mitochondria than the other rest of the cells. The most number of mitochondria that you see in this one cell in human, can anybody tell me which is the highest number of mitochondria in human cell?
Humans.
Embryonic cell has 1 lakh mitochondria. That's the highest number of mitochondria in human cell. Next is cardiac muscle neurons. High functioning cells will have more energy, so they will have more mitochondrial cells. So risk estimation is always difficult because each mitochondria will have a different number of copies of these subtle, small, tiny DNAs. So we don't know how many of them will have mutations. Based on the risk, the damage is based on the number of mitochondrial subtle DNA mutation versus the disease. The severity will vary based on the number of DNA that is having the mutation. Sometimes what happens is one single mitochondria, there will be like 100 DNA, copies DNA. If you have 20 DNAs are defective, less than 80% is normal because this condition is a heteroplasmy. So based on the quality of the mitochondria in the cell will add to the disease. So it's very difficult to predict the severity of the disease. Risk of recurrence also in mitochondrial disease. Okay, so just to summarize, whenever you see these findings, we have a possible implications for a pattern. Affected members in different generations, we say it's possibly recessive. Members in one SIP shift, so I told one generation, we can say it's most likely also recessive. Parent consanguinity increased risk for also recessive disease. Only males affected considered extinct recessive pedigree. More females than males, extinct dominant. Male to male transmission happens in the extinct, cannot happen in extinct, but probably autosomal dominant. Male have no affected descendants, mitochondrial. These are the clues. So last pedigree pattern, I just want to show you this pattern. This is called paternal imprinting pedigree. So quickly your observation for this pedigree.
Females are carriers. X-linked. It's a X-linked recessive. X-linked recessive.
Extinct.
Recessive. No, actually it's both.
Okay, I'll give a very basic, first I'll start with the base, then we'll just have a better understanding. Okay. Sir, the same. Here you can see, whenever the conditions inherit from the affected female, the individuals are having the disease. when father is affected, the individuals are becoming carriers. Exclusively affected father is not transmitting the disease. It's transmitting the defective gene, but the individuals are not affected. There's an entity called imprinting mechanism. So as I told you, 3 patterns. As I told you, we have 2 copies, always we require them. There are genes of is enough. There are genes which only express where it is coming from the father or it is coming from the mother. In this case, the genes which are coming from the father is silent. We call this as a paternal imprint. So before getting to this, I want to show this pattern. Hope you all understand this. It's a small cell and the egg cell. All of us started like this. You can have the sperm cell will have the paternal imprint, father's imprint. It's like a chapa. It's like a seed, the gene. Mother will have maternal imprint. Then individual will have the rectus. In the gonads, what happens is, again in the father, the gonads, both the maternal, paternal gene imprints are erased. And they re-established in the gamete. So, if you see here, father has both mother's copy and father's copy. But when you see the gametes, he has both paternal imprints. You can see here, this blue line is only here. You don't see the pink one, which is a maternal imprint is missing here. So this is what we call it as the molecular memory. Somehow the molecules from the father remember and establish as a paternal imprint. Likewise for the mother, you can see mother also having one copy from the father, one copy from the mother. But when in the gametes, they get erased and re-established as though mother has only the maternal copy. So this happens only in 2% of our genes. So we have about 20,000 coding genes. That 2% is roughly 200 to 400 genes behave differently based on the origin of the parent. We call this as an imprinting mechanism, imprinting phenomenon. Okay, so just to show you here, so if you take any copy here, this is a normal copy. All of us will have maternal copy, paternal copy. Many times we require both the copies to express. Sometimes half copy is enough. That predominantly, sometimes what happens is the maternal copy is expressed. The same gene on the paternal copy is silent, it's not expressed. Likewise, the paternal copy on the mother's chromosome is silent, father's copy is expressed. So this is called imprinting. Okay? You can see here what this is a normal pattern. What happened here? There's a difference here. Can anybody figure out?
In the father, the mother's copy is not expressed.
In the father,
In the mother.
here you can see both are maternal copies, and here it is both are paternal copies. So very rare entity where sometimes you get both the copies from one parent. We call it as a uniparent lysomy. So this is where the imprinting mechanism is altered. So just what you have to understand is some genes they behave differently based on the origin of the parent. Okay? That can change when you have a rare scenario like this. Both the copies coming from one parent, either from the mother or from the father. There is a change in the imprint. Okay? So here if you see this particular pattern, as I told you, the imprint is, say for example, father's genes are silent. When father's genes are silent, the father's genes which are inherited from the defective father here, what happens is the father's genes they get erased and they establish as a father's paternal copy. When the paternal copy is given, child will not get affected. When paternal copy, who is having the mother's copy, which is a defective here, they establish in the child and they get affected. So whenever you see mother is transmitting the defective gene, the the child is affected because it's a maternally they are expressing. Maternal copy is silent. So in the next example also you can see here. So same pattern, but you can see there's a difference here. Whenever the affected female is transmitting, they are not expressing. When affected father is transmitting the gene, defective gene, they are expressing the features. So here again, it is a maternally inherited, maternal imprint. So whenever the mother's genes are given, they are anyways quiet genes, they are silent genes, so they will not express the features. When father's genes are not quiet, whenever they are inherited, they manifest the disease. So this is called the maternal imprint. Imprint is usually managed by two phenomena. One is called methylation process, the other one is acetylation process. Whenever the methyl group won't bind to the gene will not express. When there is an acetyl group binds, genes will express. So this mechanism what is important for imprinting mechanism. So defect in this will usually have imprinting inheritance pattern. Usually we see this type of patterns in conditions like parallel Jeevan. We have a list of diseases that follow this pattern. So one more pedigree I want to show you. Can quickly anybody comment on this pedigree? Who are all affected? Who are all carriers? So this is a child who came to the clinic with a feature suggestive of Down syndrome. So as usual, we do a karyotype to see what is the pattern of the chromosome. So we figured out that she's having a Robertson translocation between 14 and 21. So you can see here, all are in pairs. How many pairs of chromosomes are here? 23 sets. All pairs. Till here 22 pair. This one 23rd pair. So 46 chromosomes are there. One small difference you can see here is the. So there is an extra chromosome which is actually chromosome 21 moved from here on. We call this pattern as a Robertson translocation. One of the findings that we see in children with Down syndrome, 2 to 3% will have translocation Down syndrome. When we find this kind of abnormality, okay, so you have 2 copies here, one more extra copy here, 3 copies of 21 Down syndrome. When we check the father and mother, father's chromosomes normal, mother had, can you see here, 121 is missing, then is actually shifted on to 14 chromosome. So child inherited 1421 from the mother, 121 from the mother, 121 from the father, and 114 from the father. So all put together, child became trisomy 21. Mother is a carrier.
Mother was not concerned.
Mother is a carrier. Chromosome wise, she is having a defect, but she is a balanced carrier. So whenever we have this, you can see here the pedigree. Mother inherited, she gave this 1421 and 121 from the mother and 121 which is given from the father and 14 from the father. So child got trisomy, mother is a balanced carrier. So what happened is after constructing the pedigree counseling the family, mother said that she had a sister who recently married. And then they told them that there's a possibility that these conditions can inherit. So we are, based on our counseling, she brought the sister also. We did a karyotype for her. She found to have similar translocation. So counseling and careful construction of pedigree helps in prevention of another baby with the same chromosome. So this is one of those examples I thought about here. This is a chromosomal pedigree. You don't see any dominant recessive pattern. It's one individual. So what happened is if this mother conceive again, for her the risk of having another baby with Down syndrome is high. So usually what is the risk? Mother is carrier. Actually, there's two different risks that we can give. When father is a carrier, the risk is less. Mother is a carrier, risk is more. The mother is carrier for this transformation. For her, there's 10 to 15%. The father is a carrier, 2 to 4%. Why? Because of the size of the sperm cell. The sperm cell head is very small, it cannot accommodate bigger chromosomal transformations. So usually they end up as agispermia or oligospermia. Whereas mother egg size is big, she can accommodate bigger rearrangements. That's the reason why mother has more risk than the father. So, and self-evaluation is not important. Quickly, anybody comment on this pedigree? What is the risk of, what is the pattern of inheritance? And what is the risk? X-linked dominant. Very good. What is the risk of recurrence? Risk of recurrence. If the, say for example, this lady is 50% males lethal, 50% females of
Affected, not carriers.
Okay. So actually, one case is that we have 15 minutes time. I thought I want to give this case. This is where I personally came to learn about linkage and the significance of constructing pedigree. I thought I want to give this example. So this is a very old story, 2010. Female, pregnant lady approached us when she was just 8 weeks pregnant. Her history is like this. Non-consanguineous marriage. She already lost her brother with a suspected DNA. Suspected. Clinical features suggestive of DMD, no testing done. Around 30 years, he died. No DNA saved, no test done. Clinically evaluated as mother already had a normal female child, 3-year-old, and she's 2010, she came to us with the pregnancy, 8 weeks pregnant. She wanted to know what is her risk of having baby. So this is where we all started. Before she approached us, she already did a test called carrier test. Can anybody quickly tell me what is the DMD condition can follow which pattern of inheritance? Recessive. So females carrier, males affect 50% of males will be affected, 50% of females are carriers. So she first wanted to check her DMD carrier status. When she did a test called DMD gene deletion duplication testing, and for our surprise, the test came negative. So if you know DMD genetic basis, 60% of the DMD gene will have usually will undergo deletion duplications. That's one of the causes for DMD, 60 to 60% roughly. Rest of 30%, 30 to 40%, they will have point mutations in the DMD gene. So what is point mutation? Anybody? Signal nucleotide alteration in the DNA. So especially in the DMD gene, which is located on the X chromosome PR1. This is the exact address of DMD gene. The females will have 2 copies, males will have 1 copy. So defect in this gene males cannot able to walk. At around 2 years, they lose all locomotor function. After 11, 12 years, they will become wheelchair bound. At 12 years, they will have a cardiac issue. They usually cannot survive after 20 years. So that's the usual part. So this guy died. So we have carrier status done negative. So one more information is the grandmother also underwent carrier testing. Both of them are negative for 60% of deletion duplication. Then what's the next possibility? We have to check for point mutations. 30 to 40% of point mutations. So when we inquired all labs in India, there are no single lab that kind of doing DNA sequencing. Then we emailed multiple research institutions in abroad. Only one lab in the entire world we got a reply from Guy's Hospital, London. This doctor by name Ellis Davis. He's a pediatric geneticist. So he replied us saying that we can do DMD gene sequencing. So okay, then what we did is we sent the sample of the mother to Ellis Davis lab, and they did DNA sequencing. He also asked us to send the mother and grandmother sample because it's an extinct recessive disease. So that time each test costs for 1 lakh or something. So 2 lakhs we spent. We got our data, and the data is a VUS, variant of uncertain significance. They found something, but it is for the first time they not able to correlate the clinical features with the variant. Okay, so we got again one more person. So what they advised us is, okay, let's check the gender of this fetus. If it is a female, 50% carrier. If it is a male, 50% affected. So to our surprise, they have done ultrasound, they did a CVS. Child is a male. Male. 50% chance of having this boy with a baby. Then what we did is we sent the DNA sample of the child and also other individuals, male individuals in the maternal side. So we got the sample of the mother's brothers, mother's brother. They were alike. So 2, 3 males we sent. None of them have genetic disease like DMD, but we want to know if they carry the VUS mutations. That means it is not at all going to cause the disease. So what happened is VUS was found only in the grandmother. None of these males have this VUS variant. The DNA point mutation is not. So we verified this baby's male child's DMD gene, and to our surprise, what happened is he inherited the X chromosome. This X chromosome, one comes from the grandmother, one comes from the grandfather, and this boy luckily inherited this X chromosome from the grandfather, and one Y chromosome from the X chromosome from the grandfather to mother, mother to child. So because this X chromosome is not having this VUS, they decided that they want to continue. Then after 3 years they called us, 2013, they said the boy is built. So just the technology and properly evaluating the family members made a proper decision for the family. So this is where we learned that two things I learned. One is construction of the pedigree and who is inheriting what type of genes. Number two, there's a phenomenon called linkage analysis. So there are some unknown disease like this, unknown genetic basis, where they use this process called linkage analysis. So through the technology and the proper construction of pedigree, we were able to say this. This is one of the success stories I thought which could only possible because of the technology and the proper evaluation of the family. So with this, I thought I'd stop here, and if you have any questions, we can have. And before we leave, you have to take the post test. Same questions.
We need to give some time for revision of the notes.
Definitely.
So this is 2010. What's the scenario now, sir?